So my name is Hugo Bellen. I'm the Vice President of the Genetic Society of America. And today I'll have the honor of presenting 3 awards on behalf of the GSA. So the first goes to Julie Anger of Cambridge University, who's receiving the 2020 George Beeville Award. And the Beadle Award recognizes a significant sustained service to the genetics community that goes beyond an exemplary individual research career. The 2020 award honors Julie's development of critical roles and key data sets for genetic studies and transcriptional regulation in C. elegans and beyond. Julie is, of course, a distinguished researcher in the areas of spindle orientation and cell polarity, and more recently of chromatin and gene expression. But beyond her outstanding research program, she has also built tools that have supported a huge range of discoveries by others in areas spanning biology. In particular, while screening for genes involved in worm early embryonic development and cell polarity, her lab developed a genome-wide RNAi feeding library and distributed it freely. This turned out to be a widely useful tool for the worm community. And this has really had a major impact in the C. elegans community, and her vision accelerated a stunning swath of C. elegans research. This accomplishment is simple to state, but has had huge impact on the community. And this is one of the statements of Paul Sternberg in his letter. Since then, her group has been responsible for yet more crucial community resources, this time related to chromatin and gene expression. Julie played an important role in the Modern Code project, an effort to provide an encyclopedic knowledge of the genomic functional elements in flies and worms. The Erringer lab made the first genome-wide map of histone modification in C. elegans and generated many additional chromatin mapping datasets. They also painstakingly created genome-wide maps of C. elegans promoters and enhancers across the development and aging. This was a big challenge because most C. elegans genes are transpliced, meaning that promoter locations can't be inferred from the beginning of the messenger RNA sequence. So, supplementing these resources, the Erringer lab produced Beat software to normalize ChIP-seq data and seq plots to visualize diverse genomic data. Finally, Julie is an highly active leader in the C. elegans community, serving on Worm-Based Scientific Advisory Board, and also as a co-organizer of the International C. elegans Conference. Please join me in saying thanks to Julie for her many impact contributions to the field. Julie?
Thank you, Hugo, very much for that kind introduction. I have to say I'm really honored to have been selected to receive the Beadle Award, especially because John Sulston and Bob Waterston, 2 of my worm heroes, were the second recipients of that prize in the year 2000. And when I was a postdoc in Cambridge, I was really inspired by John Sulston and other colleagues when we used to go to the pub on Friday evenings and we discussed crazy big ideas like the Worm Genome Project and the Human Genome Project, and discussed how these big ideas could be possible and how they could be doable. And that was really inspiring and motivating for me for the rest of my career. More importantly, I really want to recognize and thank the current and past members of my lab, because the award really directly reflects their amazing contributions. And I want to say it's been hugely rewarding to me to see the great science that our work has facilitated, and I hope to will carry on. So just to conclude, thank you very much to the GSA, and thank you again to everyone who's contributed to the lab's work, because this award is really for you. So thanks.
Thank you. Is the sound back on?
Yes, we can hear you.
All right. So now we turn to the 2020 Thomas Hunt Morgan Medal, which is presented to Jerry Fink of MIT for the discovery of principles central to genome organization and regulation in eukaryotic cells. The Morgan Medal is awarded to GSA members for lifetime achievement in the field of genetics. And Jerry transformed the field of yeast genetics in 1977 by developing a method for yeast transformation. This gave yeast researchers the power to target and edit genes and paved the way for many biotechnological and medical applications. Finck's group soon used transformation to show that yeast did not arrange their genes into bacterial-style operons. And they then went on to pursue a wide range of questions, making discoveries that are far too numerous to list. However, they investigated amino acid biosynthesis and regulation, MAP kinase signaling, antisense transcription, transposable elements, translational regulation, and much more. Some highlights include the discovery that many genes regulate the amino acid biosynthesis are controlled by a single master regulator, GCN4. They also showed that yeast transposable elements move around the genome by retrotransposition via an RNA intermediate. They investigated mechanisms by which fungi are detected by the immune system and how they avoid detection, and the regulation of the switch to virulent growth modes, work which has major clinical implications. Jerry also played a role in helping establish Arabidopsis as a model organism. I was surprised to learn this. Like his research, Jerry's service has been very prominent. Of course, he's a highly successful mentor and teacher. In 1970, Jerry co-founded the influential yeast genetics course at Cold Spring Harbor and taught it for 17 years, sharing his knowledge widely and seeding the yeast community. Like Finck's lab members, many of the attendees went on to highly successful scientific careers, including 28 members of the National Academy and 3 Nobel laureates. After the 2001 anthrax attacks, Finck chaired a National Research Council Committee on Bioterrorism. The product known as the Finck Report has guided policymakers about how to prevent destructive misuse of biotechnology while enabling important fundamental and applied research to continue. In the area of COVID-19, this might be still a useful report. For these accomplishments and your many contributions to the field, it is a pleasure to present you with the Thomas Hunt Morgan Award. In essence, from a famous fly biologist to a famous yeast biologist. Thank you.
Thank you, Hugo, and to the T.H. Morgan Medal Selection Committee of the GSA. This medal has personal significance because I am figuratively T.H. Morgan's scientific great-grandchild. I was first introduced to the excitement and lure of genetics research as an undergraduate by Philip Ives, who was a graduate student of A.H. Sturtevant, one of Morgan's students. This lineage transmitted to me 2 aspects of the Morgan School: the importance of teaching genetics as a way to transmit scientific reasoning, and the unique insights that genetics imparts to the understanding of biological phenomena. Morgan won the Nobel Prize in 1933, but he's best known not for his own experiments, but rather for those of his celebrated students, Sturtevant, Muller, and Bridges, whose careers he promoted. I have tried to keep this tradition alive in my own lab by fostering my students' careers and passing down to them my own idiosyncratic vision and style. My students have been successful, and they themselves have transmitted this cultural inheritance to their own students. I'm pleased to share this honor with my friend, colleague, and teacher, David Botstein. David and I have not only collaborated, both in research and teaching, but we share the same vision of the Morgan legacy. David and I created the first graduate course in genetics at MIT. We attended each other's lectures, and the experience was extraordinary. David is an animated and inspiring lecturer. But most important for me as a, quote, student in his audience, I saw genetics through his prism, and the experience was transformative. I also want to acknowledge my debt to my graduate professor, Norman Giles, and my postdoctoral advisor, Bruce Ames, both of whom are former Morgan laureates. I thank you for the honor and recognition of the T.H. Morgan Award and applaud the great contribution of the Genetic Society of America to science.
We now need to move to the next. Can you see the screen? All right.
I have to share one more time, Hugo.
I have to share one more time. Every time we unshare. Are we shared?
Sure are. You're good to go.
All right. So next we have someone who's being honored not once but twice at this conference. Bonnie Bassler of Princeton University and an HHMI investigator is the recipient of both the 2020 GSA medal and the 2020 Gruber Prize in Genetics. She will be presenting the Gruber lecture tomorrow. The GSA medal honors an individual member of the Society for outstanding contributions in the field of genetics for the last 15 years. Bonnie is recognized for her groundbreaking and influential discoveries regarding bacterial chemical communication and regulation of group behaviors. Her lab was the first to discover that bacteria use multiple chemical signals to communicate. In a process called quorum sensing, Vibrio cells release signaling molecules that they can also detect, allowing them to sense the presence of others in their vicinity by detecting the accumulated signal. Armed with this information, populations of bacteria are able to regulate their behavior on a community-wide scale. The Bassler lab has explored the language of bacteria using really a comprehensive suite of tools, usually beginning with genetics, biochemistry, then chemistry, structural biology, biophysics, bioinformatics, modeling, and engineering. They have identified a variety of chemical signals, their corresponding signaling pathways, and some of the principles of communication. They found that, as well as information about cell density, autoinducers encode information about the species identity of their neighbors. These signals allow bacteria to adjust their behavior according to whether they are surrounded by friends or foes. The Bassler lab has continued to reveal the complexity of communication between mixtures of different bacterial species, viruses, and eukaryotic cells. For example, her lab has shown that a Vibrio phage can eavesdrop on bacterial quorum sensing. This ability allows the phage to time the lysis of the bacterial host at the optimal moment. I found this very cute, personally. Addressing these fundamental questions has many implications for medicine. For example, quorum sensing can control virulence in pathogenic bacteria and the formation of biofilms, suggesting that quorum sensing modulators could serve as novel therapeutics. Many of you will know Bonnie as a wonderful science communicator, an energetic leader, a supportive mentor, and an excellent teacher. In honor of all your contributions, I'm proud to present to you the 2020 GSA medal. Bonnie?
Thank you, Dr. Bellin. I'm very moved by those nice comments. And I'm delighted to receive the 2020 Genetic Society of America medal. I understand that this prize is awarded for making an important contribution to modern genetics within the last 15 years. And so on behalf of the 25 years so far of graduate students, postdoctoral researchers, and research staff who have come through the Bassler lab, I thank you. the contributions of all of those scientists over all of those years have made today possible. I'm a geneticist, and I am also a microbiologist. Today, of course, I'm speaking to you from my home office because of the ongoing COVID-19 pandemic and shutdown. It is a sobering honor to receive the Genetics Society of America Medal in this year of all years. To me, it signifies the urgency of the scientific enterprise and the pressing need to bring a wide range of approaches to the study of microbes. I thank the Society for recognizing the importance and the relevance of genetic research on microbes. As you heard, my group's research has always focused on how microbes, as tiny as they are, can have such an enormous impact on humanity and the natural world. Any microbe alone is powerless, but as their numbers increase and they communicate and coordinate their activity, their power becomes formidable. The current pandemic is a stark demonstration of that power, and on perhaps a happier note, how that power has driven the scientific and clinical communities to, like the microbes, communicate and collaborate as never before, so together we can confront the threat of COVID-19. Beyond the current crisis, and in the spirit of being forward-looking, bringing the tools of genetics to microbial research will undoubtedly provide new ways to combat infectious disease, and we will learn how to employ microbes to make us healthier, to grow our food, to clean up our pollution, and to make ingenious bio-inspired products. I'm grateful for this award, recognizing the research from the Bassler lab. My gang is not done. Our passion for microbes and for discovering what underlies their collective power will keep us pressing forward. Thank you.
All right, this ends this session.